Genetics and genomics are constantly expanding at every level: gene discovery, variant and disease associations, diagnostic technologies, and patient care pathways. At each level, we need to create checks and balances to allow for growth while also maintaining quality, and ideally, have consensus among stakeholders in the industry. Who is responsible for these goals? And how do we navigate the exponential growth in genetics and genomics? Today, we talk with Dr. Madhuri Hegde, a co-author on the 2015 Standards and Guidelines for the Interpretation of Sequence Variants, a joint consensus recommendation of the American College of Medical Genetics and Genomics and the Association for Molecular Pathology. We start out by defining what variants are and what variant classification means.
Any change in the DNA is commonly called as mutation. So we do not in clinical genetics have been using the word mutation, but that has changed with time. So a gene in which a change is happening, we call that a variant. And that's the effect on the gene, whether it could be a loss of function variant or the gain of function variant. And the interpretation of that variant for its causality of disease is variant classification.
Next, we look at who the stakeholders are.
I look at the stakeholders like an ecosystem, which is interdependent on each other. Starting with the patients, the physician. The link between the physicians and the labs very often is the genetic counselor and the laboratories itself. Then going down another layer are the companies which are developing software for variant classification, or rather automated variant classification, or even the kits and the assays that are getting approved through FDA for commonly known pathogenic variants. And then the third level being the research community, which kind of sits all around these two groups of stakeholders, because they are doing the fundamental research, identifying new genes, understanding the changes of those variants on the gene expression or ability to cause disease itself.
Dr. Hegde gives us some insight into how the American College of Medical Genetics and Genomics, or ACMG, created the 2015 classification guidelines as the scientific community learned more about the evolving world of genetics and genomics.
So all this culminated into this short time of, and I call it a short time of 2010 to 2015, where this need for new guidelines was very much needed. Era that we went from 200 genes to about 6,000 genes, which today we say that there is some way to infer or they can cause a certain different clinical condition. And with all this happening, the difficult part of it was that how do you make sense of those 200 genes, well characterized, well assessed, still having difficulty every now and then in interpretation, and then this whole new list of genes where in the research community or the publications were very few publications, at times a single publication on a single gene, and how to bring that all together and interpret it. But it actually took us 2 full years to get these guidelines together. And there was a reason for that, because we were writing these guidelines in the middle of implementation of new technology, such as next generation sequencing, and going from panels to exomes and genomes, and a debate of what we are going to do in a clinical laboratory. Now, another things which is very important to remember is that the guidelines were meant as a generic guideline. At a very high level, they are the umbrella guidelines to use when going and drilling down into different genes, right? American College of Medical Genetics and Genomics has a global impact, and people look up to ACMG to give those guidelines out. I think this, just overall, if you had to grade the guidelines, that they have been a huge success.
We also discuss some of the hopes Dr. Hegde has for the next guideline update from ACMG.
I think achieving consensus is not an easy thing. I think that is probably the most important thing we have learned from the 2015 guideline, especially in this complex world of genetics and the new genes that are getting identified and reported. I think the next version hopefully is going to cover several things. The first thing being that there are just so many guidelines now, gene specific guidelines. We should still be able to bring these guidelines together in such a way that the umbrella guidelines can be modified to be used as a more generic guideline, so that labs don't have to sort of rely so heavily on using every single different gene guideline. Then you're starting to rely on software which are getting sort of in the market to take those single gene guidelines and accurately interpret them. The question is where that can be really done, and we should rely entirely on a software or not.
Artificial intelligence's role in variant classification has notable, critical limitations.
The last 2 years have been interesting, right, with AI, that there is so much hype about AI. And definitely AI is going to be very useful going forward. So it's not like we are all saying that, oh, this is very, very dangerous and don't do it. But the human aspect of that cannot be removed, because variant classification is a science, but also an art. And there is that human piece of it, which you just cannot remove. Variant classification does require human intervention. And it is not something you can rely on just looking at databases.
Given the ever-changing landscape of genetics and genomics, Dr. Hegde challenges clinicians to elevate their understanding of genetic testing and interpretation processes.
The question the genetic counselor should be asking the laboratory is, who is doing your interpretation, how you do interpretation, what data do you have access to doing interpretation. When you get a clinical report, talk to the laboratory, ask questions. What testing was done? What are the missing holes? Are there proper disclaimers or not? Absolutely critical when taking a clinical report and applying it to patient care. And it's very important to read the details at that point, because you are actually going to affect someone's life at that. I think in the whole scheme of things of genetics and genomics, we forget one fact, that we are still very much in the early days of how genetics is going to get applied to the population in general.
Dr. Hegde's vision for the future of clinical genetics and genomics is optimally propelled by collaboration.
To raise the tide, I think we're headed in that direction, definitely. But what do we need to do is collaboration. Today, no one can work in a silo. Collaboration, and education, like this podcast, is absolutely critical. And working with all agencies across the board.
Please follow the links to freely listen or view the full podcast, presented by the Center for Genomic Interpretation, a 501c3 nonprofit organization that works with precision medicine stakeholders to help them realize value and improve patient safety in clinical genetics, genomics, and precision medicine.